All right, everyone, we are going to get started. Oh, this is very nice. You can see the Q&A from up here. Okay, welcome back to day 3 of the first of many MIBI workshops. Today, we're going to be talking about cell level analysis. So we touched briefly on this yesterday, but really the whole of today. Oh, how do I get rid of that? Hold on. Sorry, we have. Nope, it's still there. There we go. Okay. Today we're going to be talking about cell level analysis. So the first day was all about designing your cohort data, generating it. Yesterday was about the first steps in analyzing it. And today is going to be really an in-depth dive on analysis. So just a brief overview of the ARC pipeline. This is the GitHub that we've put together that has all of the general spatial analysis tools in it. Nothing in here is specifically dependent on MIBI data. We've tried this on codex data, vector data, IMC. All of these tools are just broadly applicable for multiplex imaging. At the start, we have our segmentation notebook. So this is, you take your input data. This is going to give you the segmented objects that are your cells. We then have the Pixie pipeline. So Candace talked about the first part of the Pixie pipeline yesterday, which is the pixel clustering. We're then after that step is the cell clustering, which you'll hear about today. And then we have some post-clustering tasks, which is sort of like cleanup. The goal of this first sort of half of the pipeline is segmenting and clustering your data. What you get out of that is a segmentation mask with the location of every cell, as well as a cell table that has the phenotype and corresponding location of each of those cells in your image data. And so then now it's like, you're really ready to start doing spatial analysis. So that's the second half. We have spatial LDA, which you'll hear about today, k-means neighborhood analysis, and pairwise enrichment. And so, the first half is sort of like the high level processing, I would say, segmenting and phenotyping your data. And then the second half is all the analysis that you can do. So what are we going to talk about in this first session? First, you'll hear from me about segmenting your data. You'll then hear from Candace about the second half of the Pixie pipeline, which is cell clustering. And then from me again on how some of the tools that we started using to manually inspect and verify that these tasks are working correctly. Really, the goal of today is to understand how to generate cells, or the goal of the first part of today, this session, is to understand how to generate the cell level annotations that are going to feed into any of the downstream analysis that you can do either within ARC or in many of the other platforms and spatial tools that are continually being developed. As we've mentioned, there are pre-recorded talks going into more detail about many of these aspects. The one that is relevant for today is the setting up the ARC environment pre-recorded talk. This will walk you through the process of installing Docker, getting the GitHub downloaded, and making sure everything is looking good. The previous ones are much more focused on the Toffee pipeline because there's a lot more aspects of that may be confusing. This one is more straightforward, and it's just a lot of Jupyter notebooks. Okay, and with that, we'll turn it over to Noah for our first talk of the day. Great, thanks for having me. I'll be telling you about the work that we've been doing for putting together our segmentation pipeline. Okay, so an overview of what this talk will be. First, just to give you some background on segmenting image data in general. Then talking about the TissueNet and Mesmer development process and the paper we published on it, and then some key considerations for how you can actually use these tools to analyze your own data. Okay, so what's the deal with segmentation? Why does this even matter? When your image data comes out of Toffee or whatever perfect pre-processing pipeline you've developed, fundamentally, it's just a matrix. So you don't actually know where the cells are, where the stroma is, where anything is in your data. What you have is an intensity mask, basically, and you'll have different channels that are brighter and darker in different locations, but you don't actually know where the cells are. And so if you want to ask anything beyond like what's the total amount of PD1 in your image, you have to first identify where the cells are in the image. So instead you can say what T cell subsets are present there, what is the proliferation status of the tumor cells. All of these questions that are cell intrinsic require you to first identify where the cells are located. Because unlike flow data or single cell RNA sequencing data, the individual cells are not actually disaggregated as part of the analytical process. What you get out is a just continuous image. And so in order to subset that image and start asking these cell level questions, you first have to know where the cells are in the image. And so this is like the fundamental task of segmentation. It's generating this map you see on the right, which is called the segmentation mask, which identifies the individual precise location of every cell in your image. And this is what allows you to actually ask those single cell questions. So why is segmentation so important? Ideally, if our data was perfectly segmented, what we'd like to be able to do is generate a biaxial plot. And so this plot is something that you will see often in sort of flow type analyses, where you're going to put one marker on the x-axis and a different marker on the y-axis, and then each dot will be a cell, and you can just plot the expression of those two markers in the cells. And what you'd expect to see for something simple like CD4 and CD8, which are two markers that are mutually exclusively expressed on immune cells, is that some cells will only be positive for CD8, some will only be positive for CD4, and some will be positive for neither. And this is a nice validation that your processing pipeline and your staining and everything else is working correctly. However, when you don't have accurate segmentation, instead what you get is this plot on the right. And this is less satisfying to look at and also problematic. The reason this happens is because if you haven't correctly drawn the boundaries around your CD4 and your CD8 cells, and instead you have erroneously taken like a chunk of a CD4 and attached it to a CD8, you're now going to get this spillover between the assignments. And you're going to have cells that are mostly expressing CD8 also now falsely expressing a little bit of CD4, because the segmentation mask is what you actually use to figure out what the marker expression profile is of each cell, since they're not inherently disaggregated. So why is this so hard? One of the main reasons that segmentation, which sounds like a very simple concept, draw circles around some cells, has remained a challenge, is that image data is really heterogeneous. So shown here are a bunch of examples of different tissue types from different platforms. And what I hope you can appreciate is, even though each of these shows cells in tissues, the shape of the cells, the size of the cells, the density of the cells, all of these things vary dramatically. And so if you want an algorithm that's going to just tell you here's where the cells are so that you don't have to have a horde of undergrads doing it, you really need something that's going to be robust and is going to work across tissues and images that on their own, it's hard to tell that they're actually the same. The issue with image data specifically is that identifying features in the data itself that you could use for a machine learning algorithm is actually not that simple. And so people have tried a lot of different ways to take an image and extract the key important features from it. So on the right here is one approach where you have a photo on the left, and on the right, they sort of just identified all the boundaries and the lines that are in the image. So this is one common pre-processing step that, okay, probably much of the essential information in the image is present if you look at the borders. Another thing you can do is try and represent the image as like a vector space. And so you can, take dots and connect them and say, okay, this tells me something about like the shape of the image. And if I distort the image, how do the vectors get distorted? Another thing is you can like identify gradients in the image. So how does the intensity change from one point to the other? And all of these features are an attempt to quantify the important aspects of the image in a like mathematical way that you can then write down in a spreadsheet, basically saying, here are the key features of this image. And the reason you need to do that is because for traditional machine learning algorithms, what the input data that they operate on is a list of features. And so if you've worked with non-image data, you are familiar, even if you don't know it, with what these feature tables look like, because this is exactly what single cell RNA sequencing or bulk RNA sequencing or ATAC-seq or any of these other seqs output look like. You have a table where the rows here are going to be, some feature of interest, and the columns are your different samples. And so this could be gene expression, this could be peak depth, read depth, depending on what part of the process you're in. Fundamentally, though, it's already tabular data. And so you don't need to extract features from an image and then convert it to something like this. Your data is already in a format that is really amenable to machine learning. And so what this means is that you have a wide variety of different machine learning algorithms that can take this, what's called tabular data, and operate on it. And so you can try, more simple things like just linear regression, more sophisticated things like a random forest model, but the inputs to all of them are the same. What this means is that you can sort of play around with them and get your predictions, compare their accuracy, and in a very straightforward way, play around with different models. However, for image data, you don't have something like this. You have to kind of generate it yourself so that you can use these algorithms. And that's why people, have tried these like line diagrams, all this stuff. But it turns out that it's really hard to come up with good ways to extract features from images that work across images that are very different. And so this is where neural networks and deep learning come in. The key benefit of a deep learning algorithm is that it doesn't require feature extraction. It on its own will learn what the key features of the image are. And so all you have to do is give it the images and it gives you perfect predictions. So what I'm showing here is a segmentation task where it's trying to identify each of the unique objects in the image. And so what you can see is that it's identified the elephants, the people, the umbrellas, the backpacks, the little tiny strap of the backpack. And so there's been a huge amount of research from the computer science community on this task of segmentation, because this is so crucial for image analysis, for self-driving cars, for any application where you need to identify what the objects are in an image. And so these algorithms now can get really, really high accuracy where they are, basically extremely faithfully recapitulating what the specific shape of these many different objects are in different images. The key weakness of deep learning algorithms is that you need a ton of data in order to train them. So there's no free lunch. The reason that people just don't use deep learning for everything is A, because they're very computationally expensive to train, and B, you also need a lot of data to train them on. And this training data is of the form where you have the task you want the algorithm to do, and you've already figured out what the answer is on that task. So if it's segmentation, you already have the segmented cells. You're going to show it the image and ask it to predict it. And the way people across deep learning and specifically in biology previously have generated this training data is with this pretty laborious process where you would take an unannotated image. You would give it to an expert. You would ask that expert to say, like a pathologist, for example, please identify all the cells and their shapes in this image. The expert would then do that. And then you would feed that data into the model. And this is really, really slow. If you want 10 times more data, you either need 10 times more experts or for it to take 10 times longer. And either way, you need 10 times more money to pay them or 10 times more authorship spots. This is not scalable for the really, really large amounts of image data that are needed in order to train high quality deep learning models. And so what we decided to do was come up with a different approach. And so what I've described as phase one is often the only phase that goes on in constructing data sets. For us, though, this was just the first step. In phase two, we take this partially trained model. So it's not yet accurate enough to really give good predictions, but it's okay. And we're going to run it, even though it's not perfect, on some new data. What we then do is instead of asking an expert to annotate it, we crowdsource it. So we hire a bunch of people on the internet to annotate this data. But the key insight is that because they're not doing it from scratch, we don't have to train them perfectly to do every single step of annotation. All they have to do is find the obvious errors where two cells have been segmented as one cell or one cell has been segmented too small, and they just have to fix most of them. Then we have the expert come in, and now the expert has a much easier job than over here. The expert is just making sure that the crowdsourced annotations correctly fix the errors from the model. And so there's a huge narrowing of how much work the expert actually has to do. All they're doing is checking to make sure that there's no remaining errors. That data then gets added to the training data, and then we retrain the model and we start the whole loop again. And now the next time, because the model has seen more data that's been corrected by more people, it's going to do a better job of predictions, which means there's going to be fewer errors for the crowdsourced people to correct, which means there's fewer errors for them to miss, for the expert annotator to correct, which means this whole loop goes faster and faster and faster as your model gets better and better and better. And that's in stark contrast to this first phase, where no matter how good your model gets, it still takes just as long to generate the next set of training data because you're not taking advantage of the incremental progress in the model to generate this data. So in the third phase, your model now is good enough and you say, wow, these are great predictions. Now you take everyone out of the loop and you just say, okay, make some predictions. Here's the answer. So that's the goal, get to phase 3. The key insight is that the time it takes to produce annotations as you go from left to right decreases dramatically, so your throughput increases dramatically. Okay, so this is what we did. We created this data set that we call TissueNet, which is the largest data set of cell segmentations that's been assembled to date. And so shown here is just a nice little plot of all of the previous segmentations that had ever been published and what we included in TissueNet. And this was because everyone else had been doing this phase 1 exclusive process, which is really expensive. So even if you spent a lot of time, you just couldn't get that many annotations. But we were able to dramatically scale this up because we didn't have to have experts doing every part of the step. So as you can see here, one of the key considerations for deep learning models is that they are only as good as the data they're trained on. And so if you train a model exclusively on annotations from the colon, probably it's not going to do that well when it goes outside the colon. And so a key consideration for us was that both the types of microscope platforms it was trained on and the types of tissues it was trained on were as diverse as possible, so that even if your exact tissue platform combination wasn't in the training data set, the model would have seen enough similar things that it's still going to perform really well. Okay, so TissueNet training data set. Mesmer is what we're calling the model that we trained on this data set. And so I'm going to give you just a very high level overview of how the model works. The inputs are tissue image data. And so the specific requirements are a membrane or a cytoplasm marker to tell you what the shape of the cell is, and a nuclear marker to identify the nucleus. This data then gets normalized and tiled together and fed into the deep learning model. And what is this deep learning model actually doing under the hood? It's trying to predict both for the cell and for the nucleus, the center of each object and the shape of each object. And so this is what we've specifically asked the model to do. Please predict the exact center of the cell and also the shape of the cell, same thing for the nucleus. And then we can take those predictions and generate the segmentation mask that I showed earlier, where there will be a mask for the cell and a mask for the nucleus for every cell and every nucleus in the image. So one of the benefits of having this much training data is that it resulted in a model that was really accurate. So here we're comparing it to the model that was published in our first Cell paper in 2018 from Liat, and also another model that was released at about the same time. And what you can see here is that it's like the speed is equivalent, and the accuracy here, F1 goes from 0 being total disagreement and 1 being total agreement, is much, much higher. We also can evaluate it not just with F1, but with precision, with recall, with the Jaccard index, and all of these things in general tell the same story, which is that our model is substantially more accurate, and because of this massive amount of training data that it was trained on. So sometimes it's more intuitive to actually look at some images. So here we have a Vectra image of colorectal cancer, where the nuclei are stained with DAPI, and the cancer cells have their cytoplasm stained with cytokeratin. And what we can do is first say, okay, what did the ground truth human annotator say was the shape of these cells? Then we can, for each prediction, compute the log2 ratio of the size of the prediction to the size of the true cell. And so if these were perfectly in agreement, you'd expect this to be 0. If it was too large, it should be red. If it's too small, it would be blue. And we can do this for every cell in the image from Mesmer, which in general looks like it's hovering right around this band. There's definitely some cells that are red, there's some cells that are blue, but in general, the palette is like kind of this boring color, which is good. If we compare it to the other models, we don't see the same trend. So they look a lot more exciting, which in this case is bad, because many more of the cells are not in this white area. Also, some of them are just being missed completely. And so this is kind of visual representation of what does it mean if you have an F1 score difference of like 0.2, it actually does have an impact on the output data. Okay, so now here's that same plot just from Mesmer across a bunch of different tissue types. What you can see here is that for tissues that look very, very different, you still get highly accurate segmentations. One question is, okay, you have all this training data across different tissue types, and you just trained a single model on all of them. But wouldn't it be better if I wanted to eke out like the tiniest little performance gain? Couldn't I just train a model just on one tissue type and a separate model on each of them? Same thing with imaging platforms, and wouldn't that give me a boost? And in general, we see that the answer is not really. You know, it's essentially equivalent for training on everything compared to training on just one and evaluating on that same one, which means we don't have to sacrifice generalizability in order to boost the performance on a specific application. So, you know, these F1 scores are kind of hard to put in practice. How good is the model really? And so a natural question you might have is, if I had infinite resources, wouldn't it be better still just to pay some people to annotate the cells? And in general, what we found is no, actually. The model is doing as well as human annotators do on the same task. And you might say, wait a minute, the accuracy wasn't 100%. How could it be doing as well as humans? And the issue is that even humans disagree about what the correct segmentation is for these images because they're densely packed, they're overlapping. There isn't a true ground truth in these data. And so if you get a bunch of humans to annotate the same image and compare them to one another, and then you compare those humans to Mesmer, what you see is that the distribution of scores is the same. So the differences between typical humans are the same as the differences between those humans and Mesmer. We also asked some pathologists to evaluate which one do you prefer, the one from Mesmer or the one from the human, without telling the pathologist which was which. And you can see that their scores are all centered around 0, saying they didn't have a preference for the humans or the Mesmer predictions. So another validation that what we're doing is actually performing as well as humans in this task. So this is sort of our validation that this works well. How can you use Mesmer? And so we've made a number of different options available for how you can take this model and apply it. For those of you who are maybe more computationally familiar, we have, for those of you who are less computationally familiar, we've included it in our website. There's a plugin. You can put it in QPath. There's sort of just drag and drop ways to access the model. All you need is some image data. For people who want to play around with the guts a little bit more, we have Jupyter Notebooks. There's also a command line interface, and there's an executable Docker that can be plugged into pipeline managers. So there's a number of different ways that you can get started running this on your own data. If you want to just try it out, you can go to deepcell.org/predict. And for a full overview of all these different ways of using the model, you can go to the Intro to Deep Cell repo on the Van Valen Lab GitHub. Okay, for us, what are the key considerations when you're using Mesmer to segment your data? One thing to think about is what makes a good segmentation marker. We are sort of blessed with many different channels potentially that we could use for segmentation. You know, our friends doing Vectra, maybe you only have one membrane marker, so that's it. That's your only option. If you're doing MIBI or Codex or SciSif, you might have many different markers that potentially could identify the membrane. And so the question is, which of those are going to be good to use? In general, if a cell does not have a membrane marker included, Mesmer will not know what the shape of the cell is. And so if you want it to segment the cell rather than just the nucleus, you have to give it a membrane or cytoplasm marker. Otherwise, it is literally impossible for it to know what the shape of the cell is. And so you might have a mix of immune cells and tumor cells and endothelial cells. And so far, we have yet to find, the one marker to rule them all that will give you the shape of every single cell. So in practice, what we do is combine different membrane markers together. So we'll make a summed channel between CD45 and E-cadherin and maybe CD14, cytokeratin. It's really a panel-specific question. What is the combination of markers that's going to give you the best overall idea of the shape of the cell? So that sort of feeds into how you combine those together. A key consideration is that you don't want to just add every single marker together in your panel. You're like, well, this sometimes is a membrane marker. Let me just throw it in there. the result is going to be that when it's not a good membrane marker and it just stains everything, it's going to decrease the signal to noise of that summed channel. And so you're kind of striking a balance between having as many cells as possible represented with a membrane stain without diluting the contrast between the membrane and the rest of the cell. And so, adding like a histone mark to your membrane channel or DAPI is going to dramatically decrease the performance of the algorithm, because now instead of seeing a nice, clean, crisp honeycomb-like staining pattern, it's just going to be a blob everywhere. It's going to be like, well, I don't know what to do with that. So it's not the case that more is always better. You want to strike that balance between including as many markers as necessary without doing any more. Another consideration is how to run Mesmer. So I listed a bunch of these different options of how to use it. This in part will depend on sort of what your computational setup looks like in your lab. It's baked into the ARC pipeline as part of the Jupyter notebooks. But if you are going to be segmenting maybe a lot more images, or if you want mostly if you want more fine-grained control over the algorithm, then downloading the GitHub or the Docker image is going to allow you to do a little bit better. Okay, so what did I tell you today? Deep learning models perform really well when we train them on lots of data. Mesmer was trained on a lot of data, which we painstakingly generated with this, human-in-the-loop two-stage process, and it can be easily run to generate accurate segmentations on your own data. Thank you. Questions? Brian.
What
Okay, so the question was, what is the next step for Mesmer or segmentation more broadly? I think the areas where Mesmer continues to perform not as well as we would like are two. One is for really weirdly shaped cells that don't look like plump epithelial cells. So if they have like really long projections, then it's going to be conservative and just get the center part of that cell, but not necessarily the full extent. So that's one area that I think would be pretty easily fixed with just a lot more training data specifically of those weird shaped cells. It's just the case that many of the data sets we trained on did not have those. And so there's sort of a bias in the training data, and that means there's going to be a bias in the results. It's much more likely to say, I'm just going to capture the center part of the cell rather than the whole thing. The second is where it's hard to tell, even for a human, how to separate those cells. And so by definition, because of the image data we give it. And so if you have like a CD4 cell that's smacked right on top of a CD8, and CD4 and what we're giving it though is CD45, which maybe is positive for the whole thing, and it's a little bit out of focus, and it's not even for a human to be like, oh, is this 2 cells or 1 cell? I don't know. Right now, Mesmer doesn't do a good job separating those because we wouldn't do a good job separating those. I think the only way you could get better performance is if you actually looked at CD4 and CD8 while you were segmenting, but separately, and said, this half of this blob is CD4 positive, this half of this blob is CD8 positive. That tells me these should be 2 blobs. If you're only looking at a single summed membrane channel, you're never going to have that information to know for the borderline cases of is it 2 cells or is it 1 cell? I see, expression on one part that would not expect to be found with a cell that's also expressing the thing on the other part. Now I know to split this in half. That would require, though, training data where it's not just a single membrane channel. It's actually multiple different membrane channels. And the algorithm has to learn, when these membrane channels come together, it should be split apart. And so that would be basically the whole new TissueNet data set where you had, a lot of the training data in TissueNet came from not highly multiplexed imaging platforms. And so there weren't many examples of really deep panels that had this type of information. Now that there are many more commercially available multiplexed imaging platforms and people are publishing a lot of data sets, I think it would be reasonable that someone could go in and start doing this. And I think that would help to resolve some of these corner cases. Yes. Great. So the question was, if I have a data set with a lot of weird shaped images, weird shaped cells, what do I do to increase Mesmer performance? I think the first step is figuring out, do you need to do anything? It's a lot of work to train a deep learning model. And so if the accuracy is not perfect, but good enough for what you care about, I would say just keep it moving. Every part of this pipeline is imperfect in some way. And so it's very tempting to not move on until it is perfect, but then you would never publish anything. So what you need to do is think about, what are the specific questions that I want to answer with my analysis? And are the inaccuracies in the pipeline going to prevent me from answering that question? And if the answer is yes, then you either have to change your question or change the pipeline. But it shouldn't just be, oh, I looked at the images and I don't really like the output. That is not like a good platform for deciding to do a 1-year project of algorithm development. Optimistically 1 year. If you do through all your, nope, I really need to change it, then probably what would happen is you could take the Mesmer algorithm and you could do what's called fine tuning. So we have already trained the algorithm on all of this data. But what you could do is, with a smaller amount of data that you then from your data set go in, hand annotate, what are these, how do these squiggly shaped cells look? You could then briefly train the algorithm just on that subset of data, and it would hopefully just improve the performance on those cells without sacrificing the performance on the rest of them. But there's a lot of subtleties involved in how you actually do fine tuning. But that would be the approach I would take if I was like, I really need to get these cells, and hand annotate a smaller amount, not millions, but thousands or tens of thousands of cells, and then briefly fine tune the algorithm on those. Was any brain data included in the Mesmer training set? Absolutely not. So everything about TissueNet, Mesmer was like from the neck down. Segmenting in the brain is like terrible. I think even for humans, it's not clear what you should do a lot of the time. And so that was, if you're talking just about nuclei, if you want to just get the nuclei of your brain images, great, it'll work perfectly. If you want to do anything else, you're on your own, basically. You have these axonal projections that sometimes are in the 2D plane of the image, but sometimes they're going straight out of the image. There's these really, really long microglial processes. There's like all kinds of, especially if you're looking at disease, there's all kinds of other weird stuff in there. So in general, we said, we don't know what to do with that. Pass it to someone else, and just decided to be conservative. And for the brain, it will segment the nucleus, but nothing else. Okay, how does Mesmer compare to the S3 segmenter, Elastic, a bunch of other acronyms? Are there different use cases for each of the algorithms? Has anyone checked on some or many tissues? Okay, so Elastic we did compare to, I would say. So Elastic is not a deep learning model. Elastic uses random forests. And so what that is really good for is pixel level classifications of things in your image. So if you want to know, where is the tumor versus not tumor, but you just need it on a pixel level, or you're trying to identify some feature in the image, Elastic is great because in like 30 minutes, you can annotate a couple images and it'll give you pretty good performance. For individual cell level segmentations, the current non-deep learning version of Elastic is pretty subpar, and it was at the very bottom of our comparison. They are now incorporating new models into Elastic, and so I haven't personally tried out the deep learning aspect, but I would say for that use case, Elastic is really, really good for like other types of segmentation tasks, but we have found it to not work well at all for cell segmentation. For the other algorithms you mentioned, I'm not personally familiar with them. I would say the thing we found is that for all these different architectures, the setup of the deep learning models, those were much less important than the training data. So when we reached, the plot I showed you evaluating the performance of Mesmer against all these other models, when we retrained those same models on TissueNet, everyone did almost exactly the same. So it's not like we had some genius insight in Mesmer of like why it did so well. We just trained it on like 100 times more data than anyone had access to. And so that's what increased the performance. So if you have some other algorithm that you really like, TissueNet is available for academic use. You can just train it on that, and then you have a TissueNet trained version of whatever your favorite segmentation model is. Are there automatic ways to define which channels to mix for passing them as a memory marker? Nope. Right now, we just do visual inspection and expert assessment. If someone has an idea of a better way to do it, all of the code in Arc Analysis is, contribution friendly. So if you want to put something together and you're like, oh, I'm going to use some score to figure out which channels work and which ones don't, definitely reach out to us. If that would be, I think, a cool option to add in. But right now, the input is you just say, here are the channels I want to combine, and then combine them. Oh man, there's a lot. Albert says Shaq's motor neurons are a foot long, and so you're never going to segment the brain. Okay. Okay, how do you ensure that membrane staining is assigned to the right cell? So what happens after you segment your cells is the reason we're doing segmentation is to actually extract the signal of each marker in each cell. So what we want the output of segmentation to be is, okay, here's cell 1, here is the count of CD45, here's the count of CD3, here's the count of CD8, so that we can make this tabular data. If that cell is touching a tumor cell, and a lot of the markers we care about are on the membrane, you're going to get bleed over either from segmentation inaccuracies or just because it's actual tissue data, they're physically next to each other. And so what do you do when your tumor cell looks like it expresses CD45 because there's a immune cell right next to it? And so Candace is going to talk all about the work we've been doing in the next talk on that. The short answer is it's a problem, and it definitely interferes with downstream analysis. Does Mesmer split out segmentation masks or cell outlines? Yeah, I sort of colloquially refer to them as outlines, but the actual output is a integer segmentation mask where all of the pixels belonging to each cell have a unique integer assignment. If I performed not ideal background subtraction, will Mesmer segment some noise as cells? So that's a great question. If it really does look just like noise, Mesmer is not going to be super influenced by that. If it's like autofluorescence or noodles or something that maybe has more of a structure to it, and if you looked at it and didn't know it was like if you didn't know where it came from, you're like that could be a cell. Mesmer is going to be like that could be a cell. So that's kind of the gut check. If you had to know by looking at some other data source that actually this isn't a real cell and it's something else, Mesmer's not going to have access to that, so it's just going to assume it's a cell. So it really depends on what the source of noise is in your data. Okay, everyone likes the audio quality today, much better. Did you learn anything from the trained model about which markers work best? So I think one thing that we saw from the model is that if the best case scenario is you have a pure membrane marker. So this is something that is only expressed on the membrane and isn't in the cytoplasm. And the reason is because then it's much more obvious what the shape of that cell is. If you have like 5 cells all touching each other and the marker is only at the membrane, what that's going to look like is a honeycomb pattern, and that's very interpretable. If you have 5 cells all touching each other and it's a cytoplasm marker, then it's just going to look like everything that's not the nucleus is positive for that. And so then it's less clear where the boundaries between those cells should be drawn. And so the cytoplasm marker works better than not having one at all because it will at least expand the segmentations and will capture some of that. But the best case scenario is that you have something like E-cadherin or CD45. Most of the time we don't have that, and so then you get some reduced accuracy. But that's one thing that I think we realize is that if you have something like a keratin, which is going to be everywhere often, it's going to be less helpful than if you have a pure membrane marker. Okay, what about if I have necrosis and there's weird stuff in necrosis? Paraphrasing the question. Yeah, we also see weird stuff in necrosis. Sort of a philosophical question, like, is it because there's cell debris, actual cell debris that's in there, and so it's true signal, but we just don't care about it? Is it because random stuff is just sticking to necrosis? I think in whatever the answer is, it's a problem, and you don't want cell objects to come from necrosis, because then you're really going to have what Tyler likes to call party clusters of cells where they just express everything. So the approach we've taken is just manually identifying where the necrosis is in the image and then masking out that part of the image so that we have a flag saying, we could then go back and look at it if we want, but there's something in the cell table that says, by the way, this actually came from a necrotic region, so that you don't get confused.
Noah, just to clarify, so you're saying that like you run the whole image through Mesmer, and then in your cell table with the cell segment, you have it demarcated like a binary column if they're within some kind of necrosis.
Yeah, exactly. So Mike was clarifying that we don't mask before segmentation. We do everything normally. We like the whole segmentation pipeline, but then after the fact, the output is a cell table, and on that cell table, we will generate a mask that says this came from a necrotic region. So we don't have to change our pre-processing. It's just the post-processing that gets changed. Can I pass a nuclear marker as a membrane to segment only the nuclei, or should I use another method? So that's the great thing about the model, is that if there's not a membrane marker, so if you remember from one of those first images I showed, some of the cells had keratin as a membrane marker, but some of them didn't. And you don't just have to give up. The model is going to assume that either way, it'll get the nucleus. And if there's enough information to get the shape of the cell, it'll then expand that out. Otherwise, your cell segmentation will look very similar to your nuclear segmentation. So you don't have to do anything fancy and change the inputs. If you don't have a membrane marker, that's okay. You just give a blank channel for the membrane marker and supply the nucleus. Okay, thanks, Sven. Where do I post the questions? Okay, sounds like they got it. Yeah. So when the cells are tightly packed, would a nuclear envelope marker perform better than a membrane marker? I don't know if it would perform better. I would say it would probably give you very similar information. In that case, I would just look at which marker staining actually looks better. So I went on a long sabbatical to try and find a nuclear membrane marker that was robust, and ended up not doing that. So yeah, that sucked.
I think, one of the things that I remember we found with that was that, a lot of the more conventional nuclear envelope stains like laminate C, stuff like that, we actually found a pretty significant variation based on cell type. Sometimes it would give exquisite, beautiful silhouettes around the eye, but it was very cell type dependent, cell state dependent. But again, as Noah mentioned earlier, if someone finds a pan envelope marker that works really well, that'd be great to hear about.
Yeah, the issue, like Mike mentioned, that we ran into with the nuclear envelope markers is that the immunofluorescence cell culture vendor images looked great. It was like, wow, it only ever gets the nuclear envelope. That's awesome. And then you put it in tissue, and it's often just a pan-nuclear stain. So it's not actually, or it's 80% of the time a pan-nuclear stain and 20% of the time actually the nuclear envelope, exclusively envelope. So it ended up not being useful for that purpose. Okay, I'm going to stop now because we're going on too long. Thank you, everyone, for all your questions. Oh, didn't like that. Next up, we have Candace Liu, who will be telling us all about. There's a mic. Yeah, this is a big deal. We're serious.
All right. Hi, everyone. So in this next session, I will be talking about cell clustering and how we go from segmentation labels to actual cell phenotypes. Okay, so first I want to talk about why cell phenotyping is so difficult. Is it because it's in here? Oh. Oh God. My hair's in the no, it's fine. My hair's like all in the way. Okay. 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 So first I'm going to talk about why cell phenotyping is difficult in multiplex images. Then give an overview of cell clustering in the PIXI pipeline. So if you guys remember yesterday, I talked about how to go from MIBI images to pixel phenotype overlays where each color corresponds to a different pixel cluster. And then today I'm going to talk about how to go from the pixel clusters to cell clusters. And in this image, all the colors correspond to a different cell phenotype. Next, I'm going to give a comparison of cell clustering using our new pixel-based pipeline compared to the way our group and other groups do it using integrated expression. And then talk about applying pixel cluster cell clustering to your own data. And finally, give some commentary on different methods for cell enumeration. Okay, so quick recap from yesterday. So we talked about how you start with, after acquiring your multiplex imaging data, you can extract single pixel profiles and then cluster those pixels to get phenotypically relevant pixel clusters in your data. And I talked about how we can use that to quantify features that are not captured by traditional segmentation and how this can be applied across a variety of different imaging platforms. But today I'm going to be talking about this third facet here of how we can use the pixel clusters to improve our cell clusters. So yesterday I talked a lot about how there's a lot of stuff that exists outside of the cellular space, but still obviously cells are very, very important things that you want to quantify, which is why most people focus on the cell at the cell level. But this step is actually quite difficult. And so hopefully this pipeline makes it a lot easier to get to those cell phenotypes. Okay, so the paradigm for cell clustering using integrated expression, which is the way we used to do it, and a lot of groups who do multiplex imaging still do it, relies on the fact that you basically just add up all of the expression for each of your markers in your tissue. So what we do is we overlay the segmentation labels from segmentation labels onto our MIBI images, then just simply sum up the expression within each single cell. So that gets you to something like a cell table, what Noah mentioned you would also see in like CyTOF or single cell RNA sequencing, where you have all of your cells as rows and as your columns, all of the markers in your panel. And the numbers in those tables are simply the summed up or integrated expression of that marker in that cell. Then we would feed that table into some unsupervised clustering algorithm. Again, talked a lot about this yesterday, but the goal being you're just like grouping cells together that are more similar to each other. And often what we see in papers is something that looks like this, where you have these like nice discrete phenotypes where each row here is a cell phenotype and each column is one of the markers in your panel. But getting to this actually takes a significant amount of time and can seem a little bit like magic. So often what you actually see out of the first clustering step is something that looks like this. So here, every one of the columns is one of the 100 clusters that comes out of our self-organizing map, but any unsupervised clustering algorithm will give a similar output. And then each row here is a different marker in our panel. And so the takeaway here is that this heat map is pretty hard to interpret. So if we look, for instance, at like this first column on the right-hand side, it's expressing like a whole bunch of stuff. So it's like CD14, CD11C, PD1, 3, 20, 21. So it's like B cells, T cells, monocytes are like all mixed up in that cluster. So what we would normally do is look at cells that are inside of this cluster next to our MIBI images and manually try to figure out what those cells are. So by doing that, we are able to get to like the accurate cell clusters or cell phenotypes eventually, but it just takes a really long time. We also see that there are some clusters that look like they're expressing almost nothing. And so again, that takes a lot of manual work to go into the images and try to figure it out. So why is this the case? Why is cell phenotyping in images so difficult? Because in CyTOF and in single cell RNA-seq, it's not as difficult, and there are more like established ways that people do this. So an important consideration when looking at your images for cell phenotyping, but also just like in general, is that the images are 2D representations of like a very complex 3D object. And so the plane at which you section your tissue can make a really big difference at what you're looking at. So this is just an example of a dendritic cell here. So depending on how you section it, you might see signal that actually like looks like a cell, or you might see something that looks like just random objects. And this becomes more like also a more annoying problem when you have weirdly shaped cells. So this is an example of this in our real MIBI images. So this is CD11C, which is a dendritic cell marker. And on the right here, I've overlaid the segmentation borders onto the same image. So you can see in this example, CD11C, this dendritic cell looks very circular, looks like a cell. But sometimes we see something that looks more like this, where this looks like real signal because it's a very dense region of signal and it's very bright. So it's probably not noise, but it's also not really associated with a cell. So it's likely either like a dendrite or like the side of the cell or just something weird from like how you section the cell. And so this can cause a lot of issues because it kind of looks like all 3 of these cells or 4 of the cells surrounding it might have some of that signal when none of those cells are actually dendritic cells. So sort of building on that, this overlapping signal problem from neighboring cells can confound the phenotyping. So in this example here, we have tryptase, which is a mast cell marker, and it's quite bright. And so if we look at like the cell up here, for instance, there's a lot of that tryptase signal that is like overlapping into that cell. And when we're just simply summing up the expression of this marker, for example, in the cell above it, it's going to look like it's positive for tryptase just because of that signal at the border. And in single cell RNA-seq and like CyTOF, this isn't really an issue because you're looking at dissociated cells where you're actually taking a single cell and running it through whatever machine. Yeah. Okay, so to kind of get at these issues, we have developed this new cell clustering pipeline as a part of Pixie. So what we do is we take our pixel overlays from yesterday's talk, overlay our segmentation masks, and this time we're counting the number of each pixel cluster within each cell. So by doing this, we get a cell table where the rows are all the cells and each column is one of the pixel clusters. So in contrast to what the integrated expression version of this is, each column here is a pixel cluster, whereas before it was one of the markers in our panel. And then the clustering steps are very similar to what I talked about yesterday. We first cluster the cells using a self-organizing map. We then meta-cluster those clusters, and then we can manually adjust and then annotate those clusters using a GUI in our notebooks. So why do we think that this helps? So if we go back to this tryptase example, like I mentioned before, if we're just simply like adding up all of the expression of the tryptase expression in the cell above it, it'll look like it's tryptase positive because of like these pixels are very bright, but the actual number of those pixels is relatively low. So if we're just simply counting up the number of tryptase positive pixels, there shouldn't be that many of them. And the idea is that like the actual signal of the rest of the cell would drown out that like overlapping signal. So back to the lymph node data set that I talked a lot about yesterday. So if you remember, the lymph node is quite a structured tissue. So it's a nice way to kind of assess how your clustering is doing. In this pixel cluster overlay, this is like the T cell zone, the B cell zone, and the germinal center. And in this heat map, the rows are the pixel clusters, columns are the markers, and the colors correspond to the pixel phenotype map. And so now we ran this data through the cell clustering pipeline. And this is the overlay that we got. So now in these heat maps, each row is a cell phenotype and each column is one of the pixel clusters from the previous heat map. And the colors inside of the heat map correspond to like enrichment of those pixel clusters within those cell phenotypes. So hopefully it looks like we're able to still capture basic lymph node structure. So we have the T cell zone, B cell zone, FDCs marking the germinal center, and a lot of like APCs sort of scattered throughout the T cell zone. So in comparison, I ran the same data through the way we used to do it using integrated expression. And this is the heat map resulting from that. So in these heat maps, again, the rows are the cell phenotypes, but the columns here are, again, marker expression. So like the integrated marker expression for that cell. And so the most important thing to note here is that you get this large unassigned cluster. And if you look at the number of cells assigned to each of these clusters, you'll see that the unassigned cluster is actually the largest cell cluster. So under this paradigm, what we would do is we would go into this unassigned cluster and really try to manually figure out what these cells are. Again, we would get there in the end, but it would just take a long time. So to compare the output of the two ways of doing it, this is the same lens image I've been showing you. So now I'm just going to show you specific examples of how our method, I think, performs better than the way we used to do it. So in this example here, using integrated expression, this colored cell here, this is a CD8, was assigned as a CD8 T cell. But if we look at the actual CD8 signal, we can see that the cell above it has clear CD8 signal. And it's just that this overlapping signal is very, very bright into the cell below it. But it doesn't look like there's CD8 signal like around the cell. But if we look at the CD4 signal, it does look like the cell is positive for CD4, and using pixel composition, it was assigned as a CD4 T cell. So this is an example where we think pixel composition was able to sort of help with this problem of overlapping signal. In this next example here, using integrated expression, this cell was assigned as a 163 positive macrophage. And if we look at the 163 signal, we can see that there's sort of like small puncta of signal that may be from like a neighboring macrophage or just sectioning weirdly, but it's not sort of like nice membranous signal as we might expect. And if we look at the CD3 signal, it does look like it's more membranous, and it was assigned as a T cell using pixel composition. So this is an example of like a case where it's not just overlapping membrane signal that may cause issues, but also signal that may be like just weirdly in the middle of the cell or not very like constant throughout the cell that could cause issues. Okay, so this is an example of a cell that was unassigned using integrated expression, but was assigned as a CD68 positive macrophage using pixel composition. And we could see that it does have strong CD68 signal. So there are a lot of reasons why we see this sort of like unassigned cluster when using integrated expression. It could either just be lowly expressing or be expressing a lot of different markers that is like leading to a lot of just confusing the algorithm basically. Here's another example of where we have cells that were unassigned using integrated expression and were assigned as B cells and FDCs using pixel composition. And so B cells and FDCs are packed quite closely in the germinal center and in the B cell zone. So in those cases, it can be difficult for cell phenotyping as well because they're packed so closely, like Noah also kind of touched upon. You get a lot of that like, sort of like overlapping issue. Okay, so now going back to the cluster consistency score idea that I talked about yesterday. So I went into a lot more detail yesterday, but basically it's a way to allow us to quantify how reproducible our method is across different random initializations. So if you remember, a lower cluster consistency score is better because that means that that cell was almost always clustered with the same cells across different random initializations. A high score is bad, meaning that it was just all over the place, sometimes clustered with one group of cells, sometimes clustered with like another group of cells. And so we can apply the same idea at the cell level as we did to the pixels yesterday. And here I've colored the same image according to the cluster consistency score at the cell level. And quantifying it, we can see that using pixel composition, the cluster consistency score is lower, which is a good thing. I also use silhouette score to compare the two ways of doing it. Silhouette score is a commonly used metric to assess clustering in general. And it basically takes the inter-cluster distance and the intra-cluster distance, and there's an equation that you can use using those two values. So a higher silhouette score is better. It means that the clustering was tighter. So we can see that there's a significantly higher silhouette score when we use pixel composition instead of silhouette score. Okay, so I just want to reiterate the point that like the way we used to do it using integrated expression isn't wrong. Like all of the papers that did it like out of our lab and also like even now a lot of labs that do multiplex imaging will use this method. It's not incorrect. So it's like when you read these papers, it's definitely all real information. It's just that it takes a long time to get to those to get to accurate clusters. And so using pixel composition requires much fewer manual adjustments and can therefore save us a lot of time. And so this is, again, the same image I've been showing. These are the different cell phenotypes. We'd be able to get to the same phenotypes using integrated expression, but using pixel composition, this was basically right out of the box. Very few manual adjustments. Okay, so now I'm going to talk a little bit more about like the practical aspects of how to apply it to your own data. There's a very similar iterative process as for pixel clustering that I talked about yesterday, where you want to use your notebooks to figure out the clusters and then look at those clusters in something like Mantis or whatever software you like to assess how well it did. So there's a Jupyter notebook on Arc Analysis that is able to do this, and it also includes this nice GUI that I talked about yesterday for manually adjusting your clusters and annotating them. Yeah, so you want to use Mantis or whatever software you like to assess the cell clusters. The talk right after this will be all about Mantis, so stay tuned for that. And if you remember yesterday, I also talked about how at the pixel level, you have to think about like memory issues that may result from trying to cluster too many pixels at the same time. At the cell level, that's usually not a problem because you don't have nearly as many cells as you do pixels. So in all the data sets that we've looked at so far, there's not memory issues haven't been an issue at the cell level. And the final point here is that you may need to iterate between your pixel and cell clustering a few times. So for instance, if at the pixel cluster level, you had one pixel cluster that you were sort of like not sure about whether it was noise or if it was a real pixel cluster, or maybe it was expressing both like CD3 and CD20, you weren't sure which one to assign it to. At the cell level, those kind of issues may be more clear, like which it should actually be assigned to. So we find that iterating between the two can be very helpful. All right. So some tips and tricks for running both the pixel clustering and the cell clustering in Pixie. So sometimes you'll see cells that you know should be positive for a certain marker by actually looking at the MIBI images. But when you're looking at the pixel cluster overlays or at the cell cluster overlays, it looks like it's dominated by very noisy pixels. So to help with this issue, we found that smoothing our images can help a lot. So for certain markers, such as membrane tumor markers, the subcellular localization of those markers actually isn't very important. It's just that the presence of that marker is there, so we want to classify that cell as such. So this is an example. So this is ECAD, which in this data set was used to mark tumor cells. If we zoom into a specific region and overlay the cell borders, hopefully you can appreciate that the ECAD signal is very strong, right? It looks like it's marking the boundaries of the cell very well, but because these cells are quite large, there's a lot of space in the middle that is not expressing ECAD. And we don't, in this panel, there weren't markers that were specifically targeting like the nuclei of tumor cells. So as a result, if we look at the pixel cluster overlays, the middle of the cell is very dominated by just like noisy pixels. And so to get around this issue, if we smooth the ECAD signal more, then it'll look like most of the cell is positive for ECAD. So again, this is one of the things that you may want to change when you're iterating between the cell and pixel clustering, just to try to like see what channels actually need this adjustment. The second thing you may see is that markers that should only be in the nucleus seem to be expressed outside of the nucleus, and that is leading to a lot of noise and confounding your cell clustering. So an example of this is FOXP3. Like FOXP3 is a Treg transcription factor, and we know it should only be in the nucleus. But sometimes it can be, due to noise and other reasons, it can look like it's expressed outside of the nucleus. So here we can use masking, for example, applying a nuclear mask for FOXP3. So this is an example of FOXP3. You can see that there are like these bright puncta of signal that are probably the true FOXP3 signal, but it is still quite noisy. And if we just zoom into the specific region here and overlay the nuclei borders. So as Noah mentioned, the output of Mesmer gives you both the cell borders as well as the nuclei borders. So here I'm just showing the nuclei borders. And if we mask for just the nuclei, we can see that we're only keeping the FOXP3 signal that is within the nuclei. So that can help clean up some of the issues that may arise from FOXP3 just looking like it's expressed in a lot of places. Oh my God. Cool. Okay. So once we have our cell phenotypes, oftentimes the very simple thing to do is to simply count how many of each cell you have in each image. So here, for instance, each column is a different patient, and these are the different cell types that we're counting in our images. Okay, so how do we do this? I think this seems like a very, very easy thing to do, but I think there's a lot of nuance to this question that maybe some people don't think about when they're doing this. So this is just a very small schematic example of this. And so in this example, we're trying to count the macrophages. So the macrophages here are in green. So the easiest thing to do is simply count, right? There are 2 macrophages. But as you can imagine, that's probably not the best way to do it because the number of macrophages can depend highly on how much tissue is in the region you're looking at, and just the size of the image you're looking at, et cetera. So another way to do it is to use frequency. So that means dividing by the total number of cells in your image. So in this case, there are 6 total cells, so we divide by 6. And finally, another way to count your cells is by using a density measurement where you divide by the total tissue area. In this case, if it's like the tissue is like 75% of your FOV, you divide by that. Okay, so you might get the case where the tissue area stays constant, but the cell count increases due to like immune infiltration or some other process happening where the actual tissue stays the same, but you have a lot more cells. So in this case, the number of macrophages stays the same, but the frequency changes quite a bit because you have a lot more cells coming in. And the important thing to note here is that the density stays the same as well. So this is an example of a case where using either frequency or density would give you very different answers, right? In this case, it looks like if you're looking at frequency, that the macrophages are decreasing, whereas if you're looking at density, it looks like it stays the same. And so I don't think that there's it's not like one is better than the other. I think it really depends on the question that you're asking, which one you want to use. And finally, you can get the case where your tissue area decreases, but your cell count stays constant. So maybe you're just imaging a section of the tissue that has less actual area. And so then you can normalize for that by dividing by the area. So your density looks like it's changing a lot, but the frequency is staying the same because the total number of cells are staying the same. Yeah, so again, we talked a lot about this, like what is the best way of doing it and what we should just always do going forward. But I think the takeaway was that it just really, we couldn't decide which one was the best always. And so this may depend a lot on the tissue type and the biological question. So in very dense tissue, such as lymph node, where your cells are literally everywhere, it might not make as big of a difference because the total cell count and the total tissue area is probably going to stay constant. But if you have very sparse tissue where there's a lot of space and cells can infiltrate a lot and the total cell number can change quite a bit, it might make more of a difference and be more of a consideration what you do. Okay, so in summary, so Pixie is a complete pipeline for the annotation of pixel level and cell level phenotypes in your data. Cell clustering offers an improvement to the traditional cell clustering paradigm and can save you a considerable amount of time. And there are many ways to quantify cells, and there's no single approach that will work for every situation. Yeah, happy to take questions. Cool. No one cares. That's yes, Brian.
Going back to your sparse percent,
Yeah.
Can you make differences, I guess, in terms of what you actually looking to evaluate with more abundant technical?
Yeah, so I think, for example, in Tyler's DCIS, and correct me if I'm wrong, Eno or Mike, I think he did area freak normalization. Is that right? So I think
he did cell densities.
Okay. Yes, I think in that case, because it was very sparse and there was a lot of extracellular matrix, he decided that densities was better. And so doing frequency there, I think would actually have made quite a big difference because there's a lot of ECM and that would count as area, I think. But there was very few cells. And so doing a frequency would be lower. Yeah. Yeah.
So the question was, if you do integrated expression, no, there's two mics.
When did you take it?
If you just do integrated expression within the compartment that the membrane, that the marker is expressed in, so like FOX within the nucleus, CD45 in the membrane, does it alleviate some of the issues with integrated expression that you talked about?
Yeah, that's interesting. I think for the membrane, it wouldn't really help because the problem is that you get that overlapping signal from neighboring cells. So even if we were like looking at CD45, for instance, which is always usually on the membrane, even if we're integrating just in the membrane layer, that's from the cell next to it, if it's overlapping, you would still be adding up that signal from the neighboring cell. I don't think like the nuclear or cytoplasmic markers have that same issue. So I don't know if that would actually help much if we were just, for example, to like add up the FOXP3 signal in the nuclei. But I mean, it's an interesting idea and certainly easy enough to implement. Yeah. Yeah, totally. So I think that.
So the question was,
Thank you, Noah.
how do you know if you've gone too far down the blurring rabbit hole and you just really blur everything all the time? Are you going to misassign the neighboring cells?
Yeah, so I think this is where like sort of the iterative process is quite important of looking at them in Mantis before you decide what to do. So I think if you do see, if you are blurring too much and like everything is being assigned as a tumor cell, you'd be able to see that in Mantis well. I think, yeah, so I mean, in the data sets we've done that for so far, it looks like that wasn't really an issue, is that right? Like I feel like the tumor cells, I think at like maybe the tumor immune boundary, maybe it would be more of a consideration, but in our hands, I think it was not, I think the T cell signal is strong enough that it's still, yeah.
Okay, question is, how do you decide what markers to use for this? Is it just lineage markers, all the markers, a subset, and what do you think? How do you think about that?
Yeah, so we usually use just lineage markers. So it's similar to what I talked about yesterday at the cluster level or pixel cluster level. If you include the functional markers, you can just get an astronomical number of possibilities of like combinatorial ways the functional marker can be expressed on the cells. So in the PIXI pipeline, you choose the markers you want to use at the pixel cluster level, and then the pixel clusters are what you feed into the cell clustering. So the choice of the markers in this pipeline is really at the pixel cluster level.
Besides cell size, what other factors do you include in the intensity normalization? In PIXI, all we use is the cell size. We just count the number of pixel clusters in each cell and then just simply divide by cell size. I know that like in other labs, and it's been a debate whether or not transformations like ArcSignH, things like that, are necessary. In our hands, they were not necessary. So just to make it simpler, we did not include that. So just cell size for us. What's the impact of the Gaussian blur on segmentation accuracy?
So we don't Gaussian blur before segmentation, right? So I don't think that there's really a need to Gaussian blur for segmentation. So we don't do that. The model was trained on non-Gaussian blur data. So after we get the segmentation masked, then we just basically overlay it onto our images that we have Gaussian blurred.
I think that just echoes that, because that might be one point.
That just to clarify, you know, one of the nice things about having the segmentation, the pixel classification and everything sort of going down, you know, orthogonal routes is that you can perform sort of data transformations on prior to doing pixel clustering in a manner that is optimized for that. That does not have to be, in other words, so we can use the Sharp data to do the cell segmentation. And we can do, whatever gives us the most optimal pixel classification. We can do those data manipulations too, just to get those assignments without having to sort of, permanently blur the data that we're going to be using for all the downstream analyses. So I think that's kind of a nuance that's important to highlight.
Yeah, the other question that was asked is, isn't there a blur in Rosetta? And there is, but it's very small and it doesn't materially impact segmentation.
Yes.
Would comparing different downsampling sizes help for signal overlap?
So like downsampling the cells before you cluster. I think that to do that, you would have to have knowledge of what cells have that problem before you do it. And in very dense tissue, for instance, it's everywhere, right? In a lymph node, you have that like membrane overlapping problem basically in the entire tissue. So I don't know if that would help much because you would still have that same problem. In very sparse tissue where you only get that problem sometimes, maybe it would help, but again, you would have to pre-annotate your image with the areas where you think it's a problem and areas where it's not. So yeah.
Okay, last question. Do you worry about missing transitioning or transitory cells?
Yeah, I think this goes back to the question of like what markers you want to include. I think things that you may think exist on a spectrum, maybe that would be better left for functional characterization. Yeah, you can make the choice of what markers you want to include to try to address that. If you think there are like discrete states in the transition, then you can include markers for those transition states in the pixel clustering, and hopefully you'd be able to capture it. Again, it just depends on the question.
Awesome. Thanks, Candace. Okay, as expected, we are running late, but that's fine because we have some wiggle room in the schedule. You just place it in the charger and then it turns itself off. Okay, so the second to last talk of this session is going to go through the visualization tools we use to actually assess how our different pipelines are working. So I'll give you just a brief overview of why this actually is hard and deserves a whole talk, then how we use Mantis and some applications. So looking at annotated multiplexed imaging data is actually not that straightforward. If you're just looking at a single channel from a single experiment, truly any image software will work. But as you start adding more and more and more markers that you want to look at simultaneously, you either have to have like multiple windows open or try and make overlays in Photoshop. But it starts to become impractical if then it takes you a minute and a half or 2 minutes or 5 minutes to set up your workflow each time you want to look at a different image. So you can start making overlays. This is actually really helpful for actually looking at the co-expression patterns. This is sort of a bread and butter tool that we use so that rather than by eye or sometimes you'll like tab in between two images, if that's a solved problem, you can just look at multiple colors at the same time. The issue is, what if you want to look at something that isn't really a color, like a segmentation mask? If you want to say, is this, does this segmentation mask actually capture the shape of these cells? Maybe I have a bunch of different membrane markers, or I want to see if my weird squiggly looking macrophages are segmented. You want to overlay this segmentation on top of your image data. And the non-scientific image analysis, image viewers are not really built for this. And there's not a super straightforward way to do this. You can kind of hack it around by turning your segmentations into an outline and then a channel, but it's not really purpose-built. And it's impossible if you have annotations that you want to layer on top of that. There is, I promise you, no way to put this in Photoshop in a way that will be intelligible. And oftentimes there's really useful information. You just went through this whole process of segmenting and manually phenotyping and all this stuff. And you want that information to show up on your images so you can actually figure out, did this work? So people know this is a problem. And there's been a number of tools that have been developed that try and address this. So I've just put a couple here. There are undoubtedly many more. And the goal of these is to simplify the process of interacting with, and in some cases, annotating multiplexed imaging data. We have decided to use Mantis specifically for this step of our pipeline, which is, analyzing the outputs of our medium level processing to see how accurate it is. Why do we make that decision? One, it's really easy to install and use, so that's key. Everyone in the lab can use it. It's really easy to display different data modalities simultaneously. So you can have your raw channels, you can have your segmentations, you can have your annotations of different types, your cell table, all of that, you just plop it in there pretty seamlessly. And it doesn't have very specific data formats. The formats are a TIFF and a CSV. So anyone can format their data that way. It really doesn't require a lot of wrangling. And the Mantis team is extremely welcoming of suggestions. Anytime something is not even a bug, it's just like, man, it would be nice if it did this. They're like, okay, cool. And then it does that. Obviously, that will cease to happen if everyone in this workshop starts doing that. But in general, they have been very receptive to useful, constructive suggestions that we have given them. So what are more specifically the requirements for Mantis? A single directory with a subfolder for each image. And then within that subfolder, the relevant files. So if you have multiple different FOVs, you would put the single channel TIFFs within each one, and then the segmentation masks, the population masks, anything else that you want. So it's a really intuitive structure. It is already quite similar to the format that data from many different platforms comes out as. So why do we need to use Mantis? Candace talked a lot about using Pixie, and it's been really, really useful. But we often have this decision of which meta cluster should an individual cluster belong to. For the vast majority of them, I think it's pretty straightforward. But when there's this many clusters, inevitably there's going to be some that's like, oh, this one is like dim for CD4 and bright for CD20. Is that because there's really a lot more CD20, or is it because CD20 is brighter and really this should be CD4? And there's no way just looking at a heat map for you to know what the truth is about where that assignment should be. And so you really need an easy way to overlay that over your images and see which cluster does it belong to. Okay, so what does the Mantis cell clustering display look like? On the left side here are the channels that you've loaded in. So it's a really intuitive interface. You just drag and drop for what you want to look at and assign it to a color. You can scale it to get brighter or dimmer. And then on the right side here are the annotations that you're looking at. This could either be cell level annotations, pixel level annotations, pretty flexible. And in the center are the things that you're looking at. So you can toggle them on and off here, look only at specific channels. You can look only at specific annotations, and it's a really nice interface. So I'll take you through it now. Like I mentioned, you can toggle on and off. So when you toggle a marker on or a marker off, it's going to disappear or reappear. You can zoom in, and as you mouse over each of the segmentations, it will tell you what the annotation is, which is really, really useful. You can also toggle on and off entire sets of annotations, scroll around, find a different region, and then say, okay, let me mouse over these, see what they were assigned in a way that makes it really, really easy to explore your data. So just go through one more time. Toggle the actual image channels on and off. This is ECAD, which is a tumor marker. So we want to look at the ECAD positive cells. We turn all the annotations off so we can just mouse over them and see what their type is up on the left. And then, okay, now we're going to toggle just the tumors on and off and make sure, okay, it really looks like the ECAD positive cells are the only ones that are being marked as tumor positive here. We do a little scrolling around, make sure that's the case. And this is it dramatically simplifies and expedites how quickly you can quality check that your annotations are working. Here's another example. Now we're looking at CD4s and CD8s. Nope, yeah, CD8 T cells. So you can see there's a couple down here in the bottom left-hand corner. Now we're going to toggle on CD8 and say, okay, yep, it looks like these really are the only ones that are expressing CD8 in this image. That's good. Now we're going to check, okay, are they co-expressing CD45? Yep. There's other cells that are expressing CD45, but that's expected. These are the only ones that co-express. And now we're going to look at CD4 to make sure there wasn't a misassignment. And yet it looks like none of these 3 cells really express CD4. We can look at some other markers, CD14, go back to CD8 just to make sure that this all works. And you can imagine the specific things that you want to toggle on and off are going to be different depending on what you're worried about the misassignment being. But in general, it's really easy to do all this. You can look at the neighboring cells, say, okay, what are they? The assignment, these are monocytes, makes sense. They were CD14 positive. This process is flexible because whatever the specific question is, it's not like they've hard-coded in CD4 T cells. These are the annotations that come straight from Pixie or whatever clustering you're using. Not the Mantis team doesn't use Pixie specifically, but this is just very flexible. And so you can go through, make sure that the lineage defining markers that should be expressed are expressed, make sure that there's co-expression of the markers that should be co-expressed, and then also make sure that there's a lack of expression of the markers that you might be worried about misassignments between specific populations. So that's for cell clustering. Another step in our process is determining functional marker thresholds. So this, in contrast to cell lineage markers, which is something like CD4, CD8, CD20, we have functional markers that are expressed across a range of different cell types. So Ki67 is one we use to determine if a cell is proliferating or not. And if you're looking at something like flow data, you would just gate out positive and negative, maybe, or maybe you'd use a threshold. The issue is that in tissue data, it is more challenging, I think, just looking at the summary statistics to figure out what a good cut point is for positive and negative. Rather than seeing a nice, beautiful bimodal distribution that you just cut in the center, instead, it's more just like a single unimodal long-tailed distribution. And so then it's like, okay, how do I decide based on a plot like this, where to make the cut point? So on the x-axis here is the integrated intensity of the marker, and the y-axis is a histogram count, so how many cells are positive for that. And you can see that this is in CD8 T cells, this is in CD4 T cells, and this is in cytokeratin 17 positive tumor cells. And obviously, there are many more proliferating tumor cells than proliferating immune cells, but determining what counts as a proliferating tumor cell and what doesn't is not intuitive, I think, based just on this plot. You could imagine setting a higher threshold, in which case fewer cells would be positive, a lower threshold, in which case more cells would be positive. But looking just at this data, I think it would be very hard to be confident that you had really separated them accurately. So this is another thing we can use Mantis for. Before we were showing just the lineage markers. Here I'm now showing Ki67. So if we mouse over Ki67, we right now don't get any useful information about it. But if we add Ki67 to the plotting pane, now when we mouse over Ki67, it will tell us here is the integrated count of Ki67 in this specific cell. And so what that allows us to do is take kind of a survey, and based on the image, it looks pretty obvious which cells are Ki67 positive. So we don't have a super fancy solution, but what I've settled on, which works really well, is just look at the scores of the cells that are positive for a bunch of them in a bunch of images, and look at the scores for a bunch of cells that are negative, and use your internal neural network to be like, okay, this is the threshold that I'm going to set. Using the histograms is a nice guide initially to make sure you're not wildly off. So you look at the histogram and it's like, okay, it's probably around 0.01 or 0.02 or whatever it is. But then you actually look at the image data, and you're going to mouse over the examples that look obviously positive, look at the counts, look at the neighboring ones that are also positive, kind of see what the range is, and then look at the ones that are obviously negative and see what the values are there. And so far, I have found it very straightforward to set a threshold that separates the vast majority of the obvious positives and the vast majority of the obvious negatives. You can also change the scaling to look at some of these ones that initially appeared completely negative. You raise the threshold and see, okay, there's kind of a general background, but the same thing holds, that it is a pretty straightforward way to figure out what your markers are. And you can do this for all the different markers that you want to score. You just load them into this plot pane, change the channel that you're looking at, and this is a really straightforward way to address what has historically been a tricky part of our analysis pipeline. So summary, I think visual inspection of clustering, we also use this for segmentation assessment, is really crucial for QC, and not looking at the imaging data is going to really handicap your ability to find problems until much later down the road, where you now realize that some finding is actually artifactual. We happen to use Mantis for this specific part of our pipeline exclusively. It just works really well. It's really simple. The data is automatically formatted to be loaded into Mantis as part of the ARC pipeline. So we have written helper functions where it's like, okay, done with Pixie. Now there's like a format for Mantis function. It'll make a nice Mantis ready subfolder with a set of these test FOVs that Candace was talking about. Basically all of this, the pixel clustering, the segmentation assessment, the Mantis assessment is done on a small subset of your data, so you can really look through all of them and make sure. It will do that, and it's freely available at the following link from Parker. Yeah, thank you. Yes, Cameron.
Does Mantis have a mechanism for cell reassignment? Would you recommend on like cells in error where you just say 1 or 2 cells, not that many?
Wow, you and Kelsey are having a mind meld right now. So if you're looking in Mantis and you see a problem, what does that mean? It means that, let's say you're like, wow, 1/4 of these tumor cells are actually been labeled CD8 T cells. I need to reassign them. You're looking at the images of a subset of your data set. So if you fix it in this subset, what about the rest of your data set? You're not going to go in and manually change every single one. So this is used to identify problems, but then you have to go back and change your clustering to actually fix the problem. So maybe that means when you made your pixel meta cluster, you accidentally included an individual pixel cluster that should have been tumor cells into the immune cell meta cluster, or vice versa, or something somewhere went wrong. This is good for diagnosing the problem. I would not recommend using it for fixing the problem, because then you have to do it across every single image.
I think to that point, we're actually, Noah and I were actually talking about this yesterday, is that you don't, I would say for most of our cohorts, we go back and manually inspect them, use that to like looking at your most recent one. It looks like probably like 85–90%-ish of the assignments when you look at them look right.
And so I think the important thing to remember is that the goal here is not to, that the bar is not that you can look at your images in detail and not find one mistake. There will be mistakes, right? Absolutely. And so the main thing is that you want to just make sure that there aren't any sort of systematic biases in the mistakes, right? So it's to the extent that it's just getting cell type assignments sort of randomly wrong across all your different lineages, it's not going to really matter that much. Like the bigger issue is if it's routinely getting a specific subset wrong, right? And so just it's because you could, and this is one of the things with going down with these larger data sets, is that it's pragmatically, it's just not the software. Maybe it will be good enough sooner or later, but I mean, I think if there's anything that we even saw when you did the training data for Mesmer, is that 90% correct is about as good interperson agreements as you can even get, right?
Mm-hmm.
So it's sort of, so that's the other thing is that, is that you may look at it and be like, that's not correct, but if we got 10 expert observers to do it, I don't even know how good the concordance on that would be, right? So it's, I think trying to hit that level without systematic errors is a good goal. But to Noah's point, you don't want to get into the thing where you're going through and like manually correcting it. I'll talk to you in a few years. So.
Yeah, so Mike's point was that the goal isn't to get to 100% accuracy. The goal is that you want to make sure the output of the algorithm is good enough for your downstream task. And what that means is no systematic errors. So you're not always classifying when a FOXP3 cell is classified wrong, it's always a CD8. You want to make sure that the errors are fairly balanced, and that there's not a lot of them, but it does not need to get to 95 or 100% accuracy. For my data set, I found that around 90% seemed, the first pass of Pixie, probably there will be things you need to correct. So then you do go back in Pixie and correct them, not in Mantis. But the goal is not to keep redoing it until everything's perfect. The goal is to redo it until you're happy with the results and confident that downstream, oh my God, there's so many questions, won't be affected.
No, why don't you, because, just pragmatically, why don't you tell people, for instance, when you guys are doing this iterative evaluation, about how many images you're looking at out of your cohort, that kind of thing.
Yeah, so for my cohort, there are 1,200 FOVs. I picked 30 of them to do for the segmentation QC, for the pixel clustering QC, for the cell clustering QC, for the marker analysis QC. It was the same 30 for all of them. And I was probably on the more conservative side. I looked at almost every single image for every single step of this. I think you could be, it probably took like 2.5 or 3 weeks of fairly dedicated effort to make sure everything was looking good. You could have, that was to get the last 85 to 90% accuracy. You could have, if I had stopped at 85%, would have taken maybe a quarter of the time. But doing more than that would not have been useful. And I don't think it would have been possible to get much higher accuracy, even with a lot more time. So you can toggle between, hello? Oh, I clicked on something. The question was, can you look at multiple FOVs at a time? Not literally at the same time, but you at the top, there's similar to how there's a drop down for different channels. There's a drop down for different images that are in that data set. So you just change the drop down and it'll load a different image. It's not going to load side by side multiple ones, but I don't think that would be, for this case, so useful because you need to zoom in close enough anyway to see what you're doing that it's easy to just switch to a different image. Okay, there's a question about large images in Mantis. For us, this doesn't really come up since we don't do large images. I think it's either on the roadmap or currently happening that they're doing a pyramidal representation, so you can have very large images and then it'll zoom in just on part of it. But that would be a question for Mantis people.
The max right now is like 10,000 by 10,000.
Yeah, it's something like that. How long does Mantis take to load the data? It's currently much faster than it was before. I don't think it's a big bottleneck anymore. How quick is it for viewing large images? Okay. Okay, so there's a question about heterogeneity across different images. Can you really set a single threshold across everything? I think this is why we had an entire day on sample prep and making sure that that works. It is so much easier to fix your pre-analytical problems pre-analytically. So if you have differences in staining, you don't want to be fixing that computationally. You want to just not have differences in your staining. And so that's why we are so careful with doing everything in a single batch at the same time under the same conditions to, as much as possible, reduce batch effects, because then it really does throw a big wrench in your analysis. That's why we spent such a long time coming up with this new normalization scheme for the MIBI data specifically. As a result of this, I now was able to set a single threshold for PD1, PDL1, KI67, CD38, TIM3, TCF1 across my entire cohort, because the remaining differences in intensity were small enough that they didn't cause me to pick a different threshold. It could be with other imaging platforms that maybe it's not so straightforward to do this type of correction. If different days, maybe you have trying to compare different cohorts that were required on different instruments. There's all these other issues that could come up where this approach would be complicated. But that's why for things we can control, we really try and control it. Why did we choose Mantis over other options? I think because for this specific task, it seemed like at the time Mantis had what we needed, and the small things they didn't have, they very quickly implemented for us. So I think there's other functionality that is not in Mantis. QPath has really an extensive like H&E based interface. There's a lot of analysis tools built into it that are not yet built into Mantis. We are not saying you have to use Mantis for everything or even this, but for us, every single thing we want to do for QCing data is present in Mantis, and it's very, very simple. So I think that's why we chose it. Yep. Okay, great. Thank you.